Hello, everybody. It's my pleasure to present my research today. First, please allow me to share screen. Okay, so today I'm going to talk about IL-15 receptor alpha complex as a cancer immunotherapy in mice breast cancer model. As we know, breast cancer is poorly immunogenic cancer compared to other immunologic heart cancer, such like melanoma, colon cancer. Those cancers usually have larger mutation burden and have a higher response rate to the immunotherapy, such as immune checkpoint inhibitor. And immunologic cold tumor, such like breast cancer and pancreatic cancer, usually have very few mutation burden and often resistant to those immunotherapy. So there are a lot of immunotherapies actually in clinical trial have been tested, and so far only 2 antibodies or immune checkpoint inhibitor have been approved in combination with chemotherapy to treat advanced triple-negative breast cancer with PD-L1 positive. You can see here why they get approved, because compared to the PD-L1 negative and positive, it's obviously the response rate, objective response rate, very higher in the PD-L1 positive compared to the PD-L1 negatives. So this survive benefit of higher objection response get FDA approved. And just remember, those triple negative breast cancer only account roughly 15 to 20% of the breast cancer, and the PD-L1 positive roughly 40%. So overall, it's a very small percentage of breast cancer can benefit from this immunotherapy, combination immunotherapy. And then how about other therapies? So far, currently, the most still in the clinical trial. And so why we particularly interested in IL-15? As we know, IL-15, since it's discovered, it showed some advantage. It showed here the IL-15 showed very strong bioactivity, improving CD8 T cells, natural killer cells, survival, proliferation, and cytotoxic function. And also very importantly, IL-15 did not active or proliferate very potent suppressor cell, regulatory T cells. And also it did not active induce cell death of affected cytotoxic T cells. And also have a unique feature of this IL-15 called transplantation. So as you can see here, the IL-15, in order to get a function, they need associated with IL-15 receptor alpha form a complex, and this complex present on the beta gamma receptor to stimulate cell proliferation. So because of this transplantation, several IL-15 complex, all called super agonists, have been formed. It showed extreme or very higher bioactivity. And it can increase half-life also. So in this study, we are particularly look at IL-15 receptor alpha complex. First, we are also focused on the CD8 T cells. First, we want to evaluate the bioactivity of the complex. Then we'll also explore the role of T-bed in the CD8 T cell proliferation. And then we also will be evaluated its potential efficacy in mice breast cancer models. There's 2 mice breast models for either systemic treatment or local treatment. So first, you can see here, actually it's surprising at the beginning, you can see IL-15 and IL-15 complex did not show to proliferate CD8 T cells in vitro. You can see here. However, we can also notice the difference compared to the control of 15 receptor alone. The 15 or 15 receptor complex can actually improve those T cells survive. And importantly, if you look at the in vivo data, those IL-15 complex can significantly drive those CD8 T cell proliferation. After 5 days, you can see here roughly 1, 2, 3, 4, 5, 6, 7, 8 divisions. And however, in contrast to the equimolar IL-15 alone, did not show any proliferation activity. So we were wondering why IL-15 don't have in vitro bioactivity. Then we use another cytokine, gamma common receptor cytokine IL-7. IL-7, it also shows similar features. You can see here IL-7 alone also cannot proliferate T cell, CD8 T cell in vitro. However, if we combine two cytokines together, it showed modest bioactivity. Roughly 50% of the CD8 T cell divide. So then we look at the T-bed, if they can improve the CD8 T cell proliferation. So as you can see here in vitro data, first, because we know the CD8 the IL-15 cannot proliferate the CD8 in vitro alone, so we added the anti-CD3 as a stimulate T cell growth. As you can see here, T-bed knockout slightly decrease the capacity of the T cell proliferation, roughly 10%. And adding IL-15 can increase the proliferation in both wild type and the T-bed knockout cells. You can see here the increase is significant but small. And however, if you look at the in vivo data, in vivo data, the T-bed actually play a larger role in the CD8 T cell proliferation, both in the spleen and the lymph nodes. Without the T-bed, you can see compared to the wild type, the CD8 T cell significantly decrease the proliferation capacity, roughly 30%. Then we also look at the one of the CD8 T cell function marker, CXCR3, the expression seems no difference between the wild type and T-bed cells. You can see here, both in the wild type and the knockout spleen and the lymph nodes show the same level of CXCR3. So the conclusion is T-bed may play a partial role in the CD8 T cell proliferation with IL-15, and see to those CD8 T cells, the function maybe not affect. So we know the most potent suppressor of the cytotoxic T cells are T regulatory T cells. So you can see here in the in vitro culture, adding the Tregs significantly suppress the CD8 T cell growth. And you can see here also adding IL-15 seems antagonize the suppressor function. So you can see here this with IL-15 added, the suppressor function significantly diminished. You can see here they are very significant. And also importantly, the function of the CD8 T cells cytokine released in the interferon gamma, you can see here, the level also significantly increased. And at a 1:8 high ratio for the CD8 T cells, the interferon gamma level already recovered the same as a no Treg control. So with this data suggests IL-15 is very potent, can proliferate CD8 T cells and antagonize the suppression of regular T cells. So next, we wanted to look at the cancer treatment. So in this model called MMC model, it's called HER2 new positive breast cancer model, we treat with IL-15 complex systemically by intraperitoneal. 2 doses, day 5, day 10 after the tumor initiation. So first you can see here the tumor growth curve. We know that parental FBB mice, they are not transgenic, so they are naturally reject the tumor cells as a negative control. And compared to the 15 and 15 complex, you can see here 15 alone did not influence the tumor growth. However, if you look at 15 receptor complex, they are significantly slower tumor growth, roughly 1 week. And so then we look at the possible mechanism. And first we look at both CD4 and CD8 T cells in the spleen. As you can see here after treatment, so especially IL-15 complex, it showed significant increase in the CD8 T cells. However, CD4 T cells seems no effect. And then we look at the function of those splenocyte or cytokine T cell function release the interferon gamma. As you can see here, data suggests that both positive and negative, the T cell response to both positive and negative cancer cells. So they are not necessarily specific immunity. And to go further, we know in this model, the myeloid-derived suppressor cell is predominant in the blood and the spleen. So we look at this suppressor cells. As you can see here, surprisingly, IL-15, both IL-15 and IL-15 complex treatment significantly reduce the myeloid-derived suppressor cells. As you can see here, compared to, they are both significant still, but of course the level is still higher than control FBB mice. So because we see the myeloid-derived cells still have high levels, so next we want we interested if we can deplete myeloid-derived cells can actually help to treat tumor. So in this, we treat anti-GR1 antibody to deplete myeloid-derived suppressor cells. You can see have 3 dose treatment. In fact, as you can see here, for the control tumor mice, after antibody treatment, we almost deplete the myeloid-derived cells the same level as control mice, FBB mice. However, the tumor grow actually exactly same like the control. So although we significantly deplete the myeloid-derived suppressor cells, seeing no effect on the tumor cells. And also same thing true adding the antibody with IL-15 complex, seeing also no effect on tumor growth. They look like almost identical tumor growth curve. And however, the slight difference, if you look at the myeloid-derived cell depletion effect in the IL-15 complex treatment, the antibody seems no effect, cannot further decrease the myeloid-derived cells. So we don't know why. And then we also look at the CD8 T cells and the level, also no effect. So antibody depleted myeloid suppressor compared to the control or tumor seems no effect for the CD8 T cell level, even with 15 complex, also no effect on CD8 T cell level. So it suggests that in this model, CD8 T cells is a major anti-tumor cells. The myeloid suppressor cells seems not affect tumor growth in this case. And then seems the systemic treatment showed just a modest effect. So next we will look at the local treatment. In this model, we use 41 breast cancer, also very aggressive breast cancer model. And we treat 3 dose IL-15 receptor alpha DNA or with gene electron transfer in 1 week, have 3 dose. So as you can see here, the tumor growth, surprisingly, the tumor growth at the beginning is significantly slower. Then also we have roughly 25% of the animal get a tumor completely tumor-free. So those durable effects, we are very interested what's the mechanism. Then we look at those 2 mice, get their blood to check the affected memory, central memory level. You can see here both CD8, in these 2 mice, both CD8 and the CD4 T cells affected and the central memory significantly increased. So we suppose, assuming they can treatment induce immunomemory. Then go further, we look at the earlier point. After 2 days, we get the animal euthanize, have the spleen blood and look, and also tumor and liver, look at their local and systemic response. As you can see here in the systemic, in the blood and the spleen, we did not see significant increase of CD3 cells, and also for the suppressor cell, myeloid depleted suppressors also seems the same level as the control compared to the treatment. However, if you look at the local tumor, you can see after histology, the CD after treatment, the T cells actually migrate inside of the tumor. So you can see here, if no treatment, most T cells are actually outside of tumor to exclude those T cells. Then after treatment, the tumor actually migrate inside the tumor. So maybe IL-15 has been reported also as called T cell chemokine or T cell recruiter. So may actually increase the CD T cell inside the tumor. And also we look at the draining lymph nodes, as you can see here, in the draining lymph nodes, the tissue resident memory cells also increased, almost reached a statistically significant level. And also NKT cells, another cytotoxic T cell, significantly increased. So based on those data, we conclude for the local treatment, maybe the local change in the tumor microenvironment increase of T cells and the cytotoxic T cells may be more important to get rid of the tumor. So to summarize, we observed IL-15 and the 15 receptor complex significantly enhance IL-15 activity in vivo, and IL-15 can reverse regular T cell mediated suppressor function of cytotoxic T cells proliferation and interferon gamma production in vitro. IL-15 complex can also significantly expand CD T cells and enhance anti-tumor cytotoxic T cell proliferation in vivo. And systemically treat with IL-15 complex only results in a moderate anti-tumor activity, and compared to systemic treatment, intra-tumor gene electron transfer with complex or the local treatment, and exhibits a significant tumor regression and survive benefit. And our data also suggests the early increase of local cytotoxic T cells is critical. Cytotoxic cells is critical to the anti-tumor activity. And we also observe the depletion of myeloid depleted suppressor cells is ineffective to treat mouse breast cancer. So based on those data, you can see here systemic treatment, although can significantly proliferate CD8 T cells in systemically, however, they show the modest effect of the tumor growth. And on the other hand, local treatment with gene electron transfer, it showed no significant effect in the systemically, but however, it shows significant increase of the cytotoxic T cell in local in the tumor and in the draining lymph nodes. So it is, and also in the tumor, response, you can see here part of the tumor actually can get eliminated. So it suggests have some local actually treatment have some potential for further clinical application. So the next we want to look more for the local treatment and with because local treatment is still not enough, you can see here, and we focus on local treatment and also will combine with other therapy to further enhance the efficacy of the IL-15 complex. So to acknowledge, we have collaborators, Dr. Smells at the VCU and Richard Heller at the University of the South Florida. And also we, most of the animal study done by the Megan Scott, our technician, and when student actually, the Ninja just graduated, also did some T-cell analysis. This study was supported by NCI to New England and also Jeffrey Memorial Trust. Thank you for your attention.